Okay, so in the last class what we have discussed is we have started discussing the structure of a prokaryotic cell or the structure of a bacteria, right? And we were discussing about something called as the cell envelope. And what we remember about the cell envelope in case of bacteria is made up of how many layers, or it consists of how many layers? 3 layers, right? It's a capsule, the cell wall, and the plasma membrane or the cell membrane, right? As far as capsule is concerned, not necessarily each and every bacteria would be having a capsule. Instead of a capsule, they might have the slime layer also, right? So we have already completed something about discussion about the capsule, the cell membrane, so we'll be talking about cell wall. Is this visible? Yeah, please. Okay, so we are writing some informations about the cell wall. This is one information that you all know. We know the position of the cell wall, right? It's a complex semi-rigid structure. In bacteria, we see two types of cell wall. That is, we see two types of bacteria, gram-positive and gram-negative. So the cell wall of gram-positive and gram-negative differs, right? So whenever we talk about the cell wall of bacteria is made up of what? Obviously not cellulose, but it is made up of what? Peptidoglycan. So bacteria gram-positive will also be having peptidoglycan and gram-negative will also be having peptidoglycan, but what is the difference? We have a huge amount here and less amount there, right? That's one point of difference. And plus, in case of gram-negative, we see an extra layer in the cell wall, but in this case, it's a single layered, right? So because of these differences, there are and we can conclude that there are two types of cell wall. One basic difference between gram-positive and obviously there are so many differences between gram-positive and gram-negative, but one point from the point of cell wall is they have double layered, and in this case they will be having single layer, and plus the amount of peptidoglycan is more. Apart from that, you have to talk about this one, right? When we talk about the gram-positive and gram-negative, we have some special characteristics of gram-positive and special characteristics of gram-negative. Question has been asked as far as teichoic acid is concerned, the presence of teichoic acid is a characteristic of gram-positive or gram-negative. It's a characteristic of gram-positive. And we'll be writing certain points about teichoic acid also, what is the function, etc., etc. But as of now, I think this portion is understood, right? After this, okay, first write it down. So out of all the points that we have written about teichoic acid, the maximum question has been asked about this point. It is found in, it's found only in gram-positive bacteria, right? It acts as a receptor site. Is this point understood? Receptor means the area where you will receive a virus, right? Is it over? If this is over, next point would be point number 2, peptidoglycan. Over. Yeah. Okay, so we are writing something about peptidoglycan, that is the chief component of the bacterial cell wall. So peptidoglycan is made up of, or it is made up of different types of amino acids, right? The peptide part also is made up of certain types of amino acid, the glucan part also is made up of amino acid. Out of all the things that we have written, from examination point of view, what is important is only the points that we have marked, right? So the first point that is important is, what is murin? So bacterial cell wall is made of peptidoglycan, or you can call it as murin also. So that word is important. And as far as these amino acids are concerned, you don't have to by heart. One question that has been asked is, presence of DEPA is a characteristic of gram-positive or gram-negative. As I said, gram-positive and gram-negative would be having certain characteristics or unique characteristics. One characteristic that we have already studied is the presence of teichoic acid. Right? Teichoic acid is a characteristic of gram-positive and DEPA is a characteristic of gram-negative. These are the only 2 points that you will study. Right? Okay, and from this part, the full form, not necessary. You have to remember something called as NAG and NEM. Right? Presence of NAG and NEM is a characteristic of bacterial cell wall, but when we will be studying about some other forms of Monera, we'll be talking about that again. Differences between one bacteria and the other types of Monera. right? Over? Okay. So up till here, what we have completed is the cell envelope. We have talked about the structure of the bacterial cell, and all the parts we have marked it in the diagram. We have written the information also, right? The next part of the lesson, we have to study about staining in bacteria. So the heading would be staining in bacteria. Staining, S-T-A-I-N-I-N-G, staining in bacteria. It's over? Please. Okay. Before starting this, this unit, some of the points that we already know. First, as far as staining is concerned, we have the first question, why do you go for staining? So that the bacteria or the structure of the bacteria becomes clear under the microscope. If you want to study the structure, you have to give color, and then only the parts would be colorful and more observable, right? So as far as staining is concerned, different organelles, different organisms get stained by different colors, right? It is not like this that one stain is used for each and every individual or each and every organelle. Maybe you all have heard about something called as nuclear stain, right? We have something called as mitochondrial stain also, and now we'll be talking about something called as bacterial stain. Whenever we talk about bacterial stain, what is the stain that you remember? That is the color that they show, but what is the name of the stain? It is called as crystal violet, or it is called as Gram stain, right? Why is it called as Gram stain? Because based on who has founded it or discovered, Christian Gram, right? So these are the things that we already know, and apart from that, as far as staining is concerned, the staining process helps us in what? It helps us in differentiating the gram positive and the gram negative. As far as gram positive is concerned, they would be taking up the stain, they would be retaining the stain also. As far as gram negative, they would be taking up the stain, but they will not retain. There are two sentences, take the stain, retain the stain, right? What do you understand by retaining the stain? Something that does not go away even after you wash it down. That is called as retaining, right? So gram positive and gram negative, both of them will take up the stain, but once after washing, gram positive bacteria would remain what? Purple or violet, because they will retain the stain, and gram negative will become white again, right? So we'll just write the steps. I think the flowchart is understood, right? Now, obviously, you don't have to by heart the flowchart. What is the type of question that is asked from here? First, the name of the stain that has been used. Crystal violet, you have some other names also. It is also called the gentian violet. It is also called the gram stain. The names, important. Next is, are you done writing? Over? So what we have done is we have taken a bacteria, but instead of bacteria, what did we write? What we are doing in this process is we are staining a bacteria. So we could have written bacteria at the first time, but instead of bacteria, we have written a word called as smear, right? What do you understand by bacterial smear? Have you heard something called as smearing? Smearing process. What is smearing? You take a glass slide, with the top of it, another glass slide, you just slide it down. That is what is called as smearing. Why are we writing bacterial smear? Because obviously it's not possible to collect one bacteria with your hand and then you can go for staining. What you have to do is, since bacteria is present in water, you collect a drop of water, put it in a glass slide. With another glass slide, you just slide it down so that the bacteria gets stuck to the one of the glass slides, right? And then you go for the process. So once you are done with this process, the question that has been asked is, which step differentiates gram-positive and gram-negative bacteria? This step, right? Prior to this, you cannot be saying that the bacteria is positive or negative. So this step is the one that will be deciding as to which one is gram-positive and which one is gram-negative. Next question that has been asked is, as far as washing is concerned, how many times the process of washing has been used in this technique? 2 times, right? You wash it with water. Here we are writing it treating with alcohol. That means washing with alcohol also, right? So washing with water or washing with alcohol differentiates gram-positive and gram-negative. Obviously, washing with alcohol. Is the point clear? So how many times the washing process has been done? 2 times. Once, the first time you wash it with water. The second time you wash it with alcohol. Right? And those that retain the color, obviously they would be gram-positive and they would be purple-violet in color. And those that do not retain the color, they have to be re-stained. Because why are we staining? We are staining to know the parts or see the bacteria clearly. If they don't retain the color, again the same question, how will you see it? Right? So you have to use another stain, that is safranin, and because of safranin, the color of the bacteria turns pink. Over. Next. So talking about shapes of bacteria, you have already studied before. How many shapes? 4. What are the 4 shapes? Okay, what is the most common shape? What are the 4 shapes that you know? Something that's round, right? Next, bacillus, right? Comma shaped and spiral shaped. And the one that is common is, or the most common form is, the rod shaped or the bacillus form. Okay, talking about the shapes, 4 basic shapes: coccus, bacillus, vibrio, and spiral, right? As far as the examples of these are concerned, the examples are not being asked from examination point of view. Question that has been asked, maybe the question could be in the form of a diagram, and you would be asked to identify which type of a bacteria is it. If you see something that's spherical, so obviously it will be coccus. As far as the arrangement is concerned, you will be having the names, right? So monococcus, diplococcus, these are the 2 words that's very easily understood. Mono means single, diplo means double, right? 1 coccus, mono, 2 coccus staying together would be forming a diplococcus type of arrangement, right? And if you're talking about streptococcus, whenever you hear about strepto, you have to remember that the arrangement is like in the form of a chain, right? It is not like this that strepto would be used only for coccus. We might be having streptobacillus condition also. So you have to know the meaning of strepto. What is strepto? Arrangement in the form of a chain is called as strepto. Tetracoccus, again it's understood. Tetra means a collection of 4, right? Next, Staphylo again. Staphylo means what? Irregular bunch. This is a collection of exactly 4 of them. This is a collection of not a definite number, right? Irregular bunch we understood. It could be 5, it could be 10, it could be 11, etc., etc. Next we have Cercini. Cercini is a condition where you have, it forms a cube-like structure of 8 bacteria together, right? So you have the terms. So important is the terms and the diagram, right? And from these informations that we have written, a very important word. The diagram that we have drawn in our first class showing the structure of a bacteria, if you remember, we have shown the presence of flagella, right? But not necessarily each and every bacteria would be having a flagella. We see the coccus forms where the flagella is absent, right? So the question that has been asked is, coccus is a type of flagellate or aflagellate bacteria, aflagellated bacteria. Clear? Next, bacillus. Okay. Some points about bacillus. The most common shape is bacillus, that is rod shape. Flagellated or aflagellated. Generally flagellated. You cannot be saying flagellated in just one word. Why? Because we have used the word generally because we have certain forms of bacillus that does not have a flagella. An example of that could be lactobacillus, right? Bacillus in general, they will be having flagella, but obviously we have exceptions. Right? So we have used the word called as generally, flagellated. Out of all the information that we have written, just one word that is important, that is pellicid. Right? So what is pellicid? If the bacteria or the rod-shaped bacteria are arranged in the form of matchsticks. So again, this word is important. And as far as the names are concerned, I think we understood mono, diplo, strepto. What was the meaning of strepto? Arrangement in the form of a chain is called as strepto. So this is all about shapes of bacteria. The next topic would be number of flagella in bacteria. Over. Yeah. Yeah. So when we talk about classification of bacteria, you can classify bacteria under certain categories. First, on the basis of staining, we have seen there are 2 types of bacteria. On the basis of shapes, we have seen that there are 4 types of bacteria. Now, on the basis of flagella, we have around 6 types of bacteria. So on the basis of number of flagella, we have these terms: monotrichous, amphitrichous, cephalotrichous, etc. No doubt we have written the examples, but examples is something that you don't have to study. Question has never been asked as to give an example of a lophotrichous bacteria. This type of a question has not been asked up till now, right? So what is the question that could be asked is the diagrams, right? Based on the diagrams, you have to identify. And second, the sentences that goes with the diagram. For example, if the question is asked, the name of the bacteria that has a flagella at one end, that would be a monotrichous. The bacteria that has flagella on both the ends, that is amphi. The type of bacteria where it has two or more flagella from one end, that is cephalo. The type of bacteria that has two or more flagella that arises from both ends, right? Next, a type of bacteria that would be having flagella all around the body, that would be called as peritrichous. Could you write down the sentences yourselves, or I have to write it down? I think it's understood. So what you'll study the diagrams and the sentences that describes the diagram, right? Flagella at one end only, one flagella at one end, one flagella at both the ends, two or more flagella at one end, two or more flagella from both the ends, and all over the body, right? So this is all about shapes. Next, structure of flagella. Okay, so talking about the flagella in bacteria is made of flagellin protein. This is something that's very important. No doubt we are drawing the diagrams and everything, right? Since we are writing in the form of a note, obviously we have to write it down. But we have been talking about only that part, that is flagellin protein. Another important line that goes here, if you're talking about flagella, presence of flagella is the characteristic of a prokaryotic cell, or it is something that is present in eukaryotic cells also. Do eukaryotes have flagella? Obviously, eukaryotes, certain types of eukaryotes also have flagella, right? But there should be a difference between the prokaryotic flagella and the eukaryotic flagella. And what is the basic difference? The type of protein that it is made up of, right? Eukaryotic flagella is made up of tubulin protein, and prokaryotic flagella is made up of flagellin protein. So these are the two words that you have to remember, flagellin and tubulin. Is the point clear? So in case of bacteria, the flagella is made up of flagellin protein. How many parts does it have? 3 parts. I think from the diagram it's understood, right? So basal body, hook and filament, nothing in details that you have to study, right? Basal body, we understand the base of the flagella with which it is attached, the cell wall and the cell membrane, right? So as far as the cell wall and the cell membrane, the flagella gets attached with the help of certain rings. However, this part is also not very important, because nothing has been mentioned about this part in your NCERT book, no mention about the types of rings that they have. What is mentioned is just the 3 parts, basal, body, hook and the filament. So this is something that is extra just for our knowledge. The flagella remains attached to the cell wall, and some part of the flagella remains attached to the cell membrane also with the help of certain rings. How many rings do they have? 4. In gram-positive bacteria, it's only the inner pair is present. Inner and outer talks about what? The 2 layers, right? Cell wall is outer to the cell membrane. So this pair would be attached to the cell membrane, this pair would be attached to the cell wall. Is the point clear? Okay. Over? Any doubts? No. So we have talked about the structure of the bacteria. Next, the life process in bacteria. Talking about life processes, what are the things that we are going to study is? So 4 life processes: growth, respiration, nutrition and reproduction, right? So first starting with growth. Okay, talking about the first life process, that is growth, nothing is important, right? Because these are everything that is what? General knowledge. Where will you find bacteria? Almost everywhere, etc., etc. So if you're talking about the growth of the bacteria, I think we know this. This is something that you all have studied. S-shaped curve, remember we have called that to be a sigmoid curve, right? If you want to study the number of bacteria or you want to study the growth of a bacteria in a lab, what will you do? You'll take a petri dish, you'll put the bacteria there, and then you will see its reproduction, right? Okay, the first thing, what do you understand by growth of a bacteria? Growth means what? Increase. Increase in what? Increase in size, increase in length, etc., etc. If I say a bacteria is growing, what do you understand? Does that mean the bacteria is growing in size? Okay, if I say a human being is growing, if I say a tree is growing, means either the tree has increased in height or it has increased in weight, etc., etc. That is what growth is. But in case of unicellular organisms, what is growth? If I say a bacteria is growing, does that mean initially the bacteria was as small as this and gradually, since it keeps on growing, the bacteria will become big as this much? That is growth in case of unicellular organisms? No. When I say the bacteria is growing, we are not talking about the increase in size of the bacterial cell. Then what is growth in bacteria? Is the question understood? Right? So what do you, I mean, how can you say that the bacteria is growing? You have to see certain changes, right? What are the changes that you'll see? You know, if a plant grows, the most common thing is you'll see the increase in the height. What about bacteria, or what about any unicellular organisms? The growth of bacteria is equal to what? Something that you all have studied in the living world. Obviously, this part has been omitted now, right? Growth in case of unicellular organisms is equal to reproduction. Is the sentence understood? Growth in unicellular organisms is equal to reproduction. A bacteria is reproducing is equal to a bacteria is growing. A bacteria is reproducing means what? You had 1, now we have 2, now we have 4, now we have 8. The number keeps on increasing, right? So this is what is called as growth in case of unicellular organisms. In unicellular organisms, growth and reproduction means the same. In multicellular organisms, growth and reproduction means different, right? So if I say a bacteria is growing means what? You have, suppose we are talking from lab point of view, you have a petri dish, you have started your experiment with 1 bacteria, right? This bacteria will become how many? 2, then they will become 4, then they will become 8, etc. This is the growth of bacteria, right? So what we are writing here is, if you want to study the growth of bacteria, what you see, the bacteria will be, all the informations that you collect, you can put it in the form of a graph, and the graph would be S-shaped, yes or no? Why? Because initially, if you're talking about 1 bacteria, it will grow in a very slow rate. Next, it will become 2, next it will become 8, next it will become 16, and then we'll have an exponential growth, right? So this first phase would be called as what? Leg phase. What is the characteristic of a leg phase? The growth is very, very less, right? After the leg phase, you have seen this sharp increase. So this is called as log phase. Log phase is also called as exponential phase. Means this is a phase where maximum growth has taken place. Maximum multiplication of the bacteria has taken place. Is the point clear? So once it keeps on growing, it cannot be growing at constantly. At one time, it will become steady, right? And then it will bend, right? So next is stationary, and finally the bacteria will die. So if you want to study the growth of a bacteria, what you see is you collect all the information, you put the information in a form of a graph, and that graph is called as what? Sigmoid curve. Is the point clear? So the question that is asked is only on this. What type of a curve is seen in bacterial growth? Or a bacteria shows that dash type of a growth curve. It shows a sigmoid type of a growth curve. Is the point clear? Right? Nothing more than that. Right? So bacteria shows a sigmoid type of growth curve. Clear? The next life process is respiration in bacteria. Whenever we talk about respiration, how many types of respiration are there? Respiration, how many types? 2 types. What are the 2 types? You don't know the 2 types of respiration? Either the organism would be respiring dash or dash. 2 types of respiration. Heard about the words aerobic and anaerobic? Yes or no? So respiration is broadly classified under 2 categories. Either it would be aerobic or it would be anaerobic. The same classification in case of bacteria also, right? So when we talk about respiration, we see 2 types of respiration. As far as the difference is concerned, I think we know this, right? Aerobic respiration, anaerobic respiration. Respiration in presence of oxygen and respiration in absence of oxygen is called as aerobic and anaerobic. What is respiration? What happens in respiration? The food breaks down, right? Why do you respire? To get energy, right? So if that breaking down of food takes place with the help of oxygen, that process would be called as aerobic respiration. If the food that is present in the cell gets broken down without oxygen, that would be called as anaerobic respiration. So bacteria can have both the 2 forms. There are certain aerobic bacteria, there are certain anaerobic bacteria, right? Again, the aerobic and anaerobic bacteria has been classified under 2 categories. We have 2 more words, obligate and facultative. From this chart, what is the type of question that is asked? Give an example of an obligate aerobe. Give an example of an anaerobic bacteria. Give an example of an obligate anaerobic bacteria. Give an example of a facultative anaerobic bacteria. So each and every word together with the examples important. There are certain charts where we have said don't study the examples. Right? As far as shapes is concerned, obviously examples not important. As far as, coccus, bacillus, whatever we have written, right? But from this chart, the words are important, the examples are also important. Now, talking about this chart, aerobic and anaerobic are the 2 points that we have understood. In presence of oxygen, in absence of oxygen. Now we come across another 2 terms, that is obligate and facultative. What is the meaning of obligate? What is the meaning of facultative? Obligate means what? Do we understand this? Right? And facultative is the term understood? If you are strictly bounded to something, that is called as obligate. If you can keep on changing, that is called as facultative, right? So you have the first category called as obligate aerobes. Means what? They are strictly aerobic. These type of bacteria, in absence of oxygen, will never survive, right? To survive, they will always need oxygen, and so they are called as obligate aerobes. However, there are certain types of aerobic bacteria which can survive under both the conditions. They can survive in presence of oxygen, they can survive in absence of oxygen also. Whenever oxygen is present, they will be behaving as aerobic. Whenever there is lack of oxygen, they will switch over to anaerobic type of respiration. So that's the reason why they are called as facultative. Means what? If they are given a chance, they will go to anaerobic. Originally, they are aerobic, but if given a chance, they can become anaerobic also. That is the meaning of facultative. Is the point understood? Right? Similar is the explanation in this case also, right? So we have obligate anaerobes, means these bacteria cannot survive in presence of oxygen. If they have to survive, it should be an anaerobic condition only, right? But we have another group of organisms that are called as facultative aerobes. If the question is asked, what type of an organism is a facultative aerobe? Facultative aerobe is a type of aerobic bacteria or anaerobic bacteria. Obviously, it's an example of anaerobic bacteria. No doubt we have written aerobes. Is the point understood? Facultative aerobes is a type of aerobic bacteria or it's a type of anaerobic bacteria? It's a type of anaerobic bacteria. Why? Because these are strict. These are anaerobic, but provided they are supplied with oxygen, they can survive in that condition also. Clear? So this is all about respiration. The next is nutrition. Put the heading, nutrition in bacteria. Is respiration part clear? We have talked about two life processes. One is growth, and you would be studying just one point about growth. Respiration, you'll be studying all the points, right? Okay, so when we have started respiration, how many types of respiration do you know? Two types of respiration. If you talk about nutrition, how many types of nutrition are there? When we talk about respiration, there are two types of respiration. Either it would be aerobic or it would be anaerobic. If you talk about nutrition, how many types of nutrition are there? Nutrition means what? Food. Yes or no? So what would the organism do to have food? Either they will make their own food or they will take the food from someone else. We have two terms for that. What are the two terms? If an organism prepares its own food, they are called as autotrophs. And if they don't prepare their own food, they get it from somewhere else, they would be called as what? Heterotrophs. So the classification would begin from those two words only. Either they are autotrophs or they are heterotrophs. If an organism is a heterotroph, the first point that we know is they will take the food from some other source. Based on which source they take the food, we have classification of heterotrophs also, right? So any example of a heterotrophic mode of nutrition? I'm not asking an example of a heterotroph. I'm asking a heterotrophic mode of nutrition, types of heterotroph. I'm sorry? Holozoic type of nutrition. I'm talking, oh yeah, that is one type of, it's a type of nutrition, right? But if you're talking about classification of heterotrophs, we are talking from the source, from which source they will take the food. See, if the organism don't prepare its own food, either they will take the food from a living host, or they will take the food from a dead host, or they will remain in a symbiotic association. If you don't prepare your food, you take it from someone else. If I take it from a living host, then I'll be a parasite. If our organism take the food from a dead host, then it would be called as what? Saprophytes. They would be called as a saprophyte. And there are certain organisms which lives in a symbiotic association. They would be called as what? Symbionts. That is the classification of heterotrophs, right? So in case of bacteria also, we see two types, autotrophic bacteria and heterotrophic bacteria. So nutrition in bacteria, autotrophs and heterotrophs. If it is a heterotroph, it would be a parasite, saprophyte, or a symbiont. If it is an autotroph, if they prepare their own food, they would prepare their own food in presence of light or in presence of chemicals. If the food preparation is in presence of light, they will be called as photosynthetic. If they prepare their food in the form of, with the help of chemicals, they would be called as chemosynthetic. I think we understood the meaning, right? If it is photosynthetic, again, we have two classifications, photoorganotrophs and photolithotrophs, and in that case also we have two words, chemolithotrophs and chemoorganotrophs. Like chemo and photo, these are the words that we understood. Presence, prepare their own food with the help of light or prepare their own food with the help of chemicals. We have to understand the meaning between litho and organo, right? So photoorganotrophs and photolithotrophs differs in what? Differs in what? One thing, the source of electron and proton. Whenever you are talking about food preparation, then every time an organism prepares food, you need a source of electron and proton so that ATP can be formed. If you remember, you have studied photosynthesis, right? So whenever you talk about photosynthesis, you remember, yeah, we have studied about light reaction and dark reaction, right? Do you remember what is the product of light reaction? What is photosynthesis? Preparation of food, right? What is the product of photosynthesis? Obviously carbohydrates, right? But photosynthesis have been divided into two reactions, light and dark. When is food prepared? Light or dark? Food in the form of carbohydrate is the ultimate result of photosynthesis, yes or no? When is it formed? In the light reaction or it's formed in the dark reaction? It is a product of the dark reaction, it's not a product of the light reaction. So if you don't know this, how can I ask you the second question, what is the product of the light reaction? Remember something or everything over? Okay, we'll talk about that in the next class. As of now, do you remember the reaction of photosynthesis? The entire process of photosynthesis could be put under a particular reaction. Dash plus dash will give you food. What plus what gives food? Remember that, obviously you have studied, right? Something plus something will give you food. Carbon dioxide plus water will give you food. Yes or no? That's a basic reaction of photosynthesis. What happens to carbon dioxide? What happens to water? Just forget. What is the role of water in photosynthesis? Water acts as a source of electron and proton. How? We'll discuss in photosynthesis. As of now, what I want to say is whenever we have a food making process, you always require a source of electron and proton. In photosynthesis, what was the source of electron and proton? It was water. And because of that, again, ATP gets formed. Because of that, oxygen gets evolved, right? So every time someone prepares food, you need a source of electron and proton. If the source is an organic substance, it would be called as photoorganotroph. If the source is an inorganic substance like H2S, right? Electrons and proton, just understand it very simply. What was that? You don't know? Okay, fine. Okay. So, okay, understand it very simply. Whenever there's a food making process, you need a source of electron and proton. Someone has to supply electrons and protons. Why? To form ATP, right? Okay, forget. That source could be either an organic substance, it could be an inorganic substance also. Organic substance like carbohydrates, proteins, etc., etc. Inorganic substance like H2S. If the source is an inorganic substance, we use the word litho. If the source is an organic substance, we use the word organo. Is the point clear? Right? As far as these examples are concerned, this is something that we'll do it tomorrow, or maybe in the, obviously in the next class, if we have a class tomorrow. And one example of this has already been given. I think this is a word that you have understood, or you have heard it before. Methane bacteria. Remember these words, methanococcus, right? Remember that word called as, okay, forget, archaebacteria, right? We'll study then. This is all for today. Thank you.